So to begin, my project is Muscular Dystrophy through Bioinformatics. Oh yeah, my mentor was Dr. Brennan, and my co-mentor was Ms. Jo McMahon. So basically, to start off, what is muscular dystrophy? Muscular dystrophy basically is a genetic hereditary disease that causes muscles to deteriorate and waste away. And in this image, right here, basically it's just showing a muscle cell how it's supposed to look, because it's caused by the dystrophin gene. And this is how it's supposed to look, and this is how it happens in Duchenne muscular dystrophy, which I'll explain further on in the presentation. So the causes is mutated gene dystrophin, and it's found on the X chromosome. So since men only have one X chromosome, they're most common to have the disease, and when people have two, are more likely to be carriers but can't have the disease if they have two copies on two of their X chromosomes. So here is a picture of dystrophin under the twins. And some explanation. Becker's and Duchenne, which are basically the same thing, but Becker's is more of a less severe version of Duchenne, and their X-linked recessive disease, and it's caused by the mutation of the dystrophin gene, and leads to overproduction of the enzyme creatine kinase. So this picture here shows you exactly where it is positioned where the mutation actually kind of occurs. It occurs in the short arm of the X chromosome right here, and XP21.1 between XP21.3 as well. They both affect the muscle cells, similarly in the skeletal muscles and the heart. Becker's and Duchenne are a mutation, which is a single base substitution, and I'll further explain that. And Duchenne is the main time dystrophy, and Becker's is a less severe version. In this image here, it shows you the breakdown of how proteins are made. Basically, it starts off with DNA, and then it transcribes to messenger RNA, which then splices to basically get the exons, which are coded for the amino acids. And amino acids are like small pearls, and like a chain of pearls are a protein. This second image here shows you the point mutation, which I told you is a single base substitution. Right here is CAA, which codes for glutamine, which is right there. And here is UAA, which is a stop codon. So basically it changed because either one of the exons are missing or some sort of mutation happens. So that's what happens, is the mutation. In the next slide here, this is basically a DNA sequence of dystrophin. So this is a sequence for the normal. This is mutated at the other side here for you guys. I compared them, and basically, right there, the little box, are the mutation where it occurs. So GCAA, or GCA, which codes for alanine, and it basically changed to ACG, which is threonine. Okay, so the next slide shows you the normal, which is right here, and this is the mutated creatine kinase. This is the enzyme that's created by the dystrophin gene. And because of that one mutation in the gene, it went down when it was making the protein, and it caused mutation in creatine kinase. So here are the three mutations, and you can see here, so it was supposed to be glutamic acid, but some reason, because of the mutation, it became glutamine. And another one here, it was supposed to be aspartic acid, and it became glycine, and another one was supposed to be glycine, and became alanine. So this is the chromosomal location. This is the X-ray chromatography of DNA. Sorry, it's supposed to be X chromosome. But this is basically caused by mutation, like I said, by XP21.1 and XP21.3. And it codes for enzyme creatine kinase. So there are different ways of finding out if you have muscular dystrophy. So there's a CPK test, which measures the amount of CPK in the bloodstream. Basically, in muscular dystrophy, the muscles or the cells leak out CPK. So if there's like an excess amount, the doctors will be able to pick it up. In a DNA test, they will measure the number of exons. 79 exons are basically coded for muscular dystrophy. So if you're supposed to have, I think, more exons, so just like normal. Muscle biopsy, which basically where they extract muscle tissue from, and then they'll basically take it under the microscope and dye it, and you'll be able to see dystrophin, which is how it should look. An image of how dystrophin appears when seen in the muscle fibers. And this is the symptoms: muscle weakness, difficulty walking, hypertonia, which is low muscle tone. And it can lead to serious complications such as scoliosis, which is a curvature of the spine, mental impairment, and cardiomyopathy. Cardiomyopathy basically is where the heart is deteriorating the muscle. So there aren't exactly any cures for muscular dystrophy, but there are some sort of ways that they can help people cope with the disease. And so people actually go through physical therapy, actually have some limbs replaced, or in some cases have surgery. And prevalence is basically dystrophy is a rare disease, and it happens to 1 in every 544,000 people. And symptoms of this disease begin at the beginning of the age of 6, and the expectancy are like from the teenage years to mid-30s, but there are some cases where people actually survive to the age of 50. So new discoveries I have here. Basically, scientists have been making some new discoveries, and they've been using the drug Viagra because it is a PDE5 inhibitor, which is a phosphodiesterase inhibitor. It breaks a strong covalent bond, which is in the 3' carbon atom, and it also breaks the 5' sugar in DNA, which happens to be deoxyribose, and RNA is ribose. And basically, CGMP, it helps with the function with dystrophin also. So we include bioinformatics to basically have to explain what bioinformatics is. So bioinformatics is an emerging field in science which consists of biology, computer science, and information technology, and it allows researchers to interpret data such as nucleotide amino acid sequences and observe mutations in human genes. So my experiment was to find a mutation used in humans that can cause muscular dystrophy and determine and calculate a person's chances of developing disease. So I used the program on NCBI, which is BLAST, which is the basic alignment search tool. And it's basically a database which has different DNA sequences. And researchers basically enter their sample of their DNA sequence, and the tool, BLAST, basically compares it to the database. This is my research. I use the BLAST program on National Center of Biotechnology and Information website. And my mentor gave me a program called Name That Gene. And on this program, I just picked a random gene and I just copied it, pasted it into BLAST, and I'll show you what I did. Okay, so basically I had the gene and I copied it from the Word document. And we went to BLAST. We went to nucleotide BLAST. And I was supposed to copy and paste it here, and it will show you basically the research I did. It shows you the comparisons, and it also tells you the mutation, which is in dystrophin, and basically gene ID and things like that. And these are my references, my acknowledgements. To acknowledge my co-mentor, Ms. Jo McMahon, my mentor, Thomas Brennan, Eric, who's a computer technician, Ms. Gonzalez, faculty staff from UCC, Bronx Community College, Dr. Satt, Pharmacist Society, staff, and HTS members. Thank you.